really only had RNA, was using this RNA handle on this vitamin cofactor to actually grab onto it to do reactivity. Now I mentioned the word vitamin because if you actually look at your vitamins, you'll discover all sorts of things which have RNA cofactors associated with them. So there's ATP, but there's niacin. It's also found in all kingdoms of life. It also has this RNA piece slapped onto it where it doesn't particularly do much role biochemically. It is used in oxidation reduction reactions. This guy over here is coenzyme A. The vitamin that you have is pantothenic acid. If you look at your vitamin pills, you'll see this. It also has a piece of RNA slapped on the side of it. This is flavin adenine dinucleotide. There's a piece of RNA slapped onto it. It's the vitamin riboflavin. In fact, if you go through metabolism, which you have all across life, not just in you, not just in corn, but in all bacteria as well, you'll discover these RNA molecules have these RNA vitamin cofactors have RNA having no function, and the question is, why is it there? And the answer is because you can place it in the ancestor, but that implies that there was a time where these RNA handles were useful because RNA was really the only thing you had. And this was actually developed in the 1970s by some guys. We wrote a paper of it in 1989, and one of the major discoveries for which a Nobel Prize was won was that they went back and looked at the machines that make proteins. There's the machine that makes proteins. It's called a ribosome. It's got orange parts. Those are the RNA, but it also has a few proteins slapped on the outside. But the important part of that machine is it is the RNA that's making proteins. And that implies that RNA as a catalyst comes before proteins. And so this is the so-called RNA world hypothesis that has an episode of natural history where RNA is the sole genetically encoded biocatalyst. It explains the RNA vitamin cofactors with RNA handles that bind RNA enzymes. DNA, according to this story, later emerged from more stable genetic molecules. Then RNA catalysts then emerged to make proteins. There's one of them. This avoids this so-called chicken or egg problem where you need proteins to make DNA and DNA to make proteins. According to this story, RNA did both first. And for this to make sense, RNA world must have had a complex metabolism to make all these cofactors. So that's what RNA is. And so we can actually test this. So one of the hypotheses is that there is an RNA molecule able to catalyze its own replication. Here's Phil Holliger, as a former student of mine. He's now at Cambridge in the UK. He has actually made an RNA molecule that's able to catalyze the template-directed synthesis of RNA. And there it is. Okay? So you can say, okay, great, are we done? Well, if RNA were, in fact, the first Darwinian biopolymer, we should be able to get RNA out of a prebiotic soup, right? Especially since we're intelligent, right? And so I can sit there with a catalog, buy reagents, mix them in the right order, and I don't have to wait around for something to happen accidentally. And this is actually all summarized in this book. It turns out we can get the bases. We have not been able to get the RNA. There's been 60 years of failure. So now that I've explained to you why we want RNA, because it looks like the first Darwinian system, I've got to explain to you why we can't get RNA. Now, by the way, in some cases it should be easy, because the early Earth's atmosphere is carbon dioxide, dinitrogen, and water. If you put electrical discharge through that, a little bit of ultraviolet light, you will get out formaldehyde, glycolaldehyde, hydrogen cyanide, and cyanamide. Now, formaldehyde is C1H2O1, glycolaldehyde is C2H4O2, ribose, by the way, is C5H2O5. And so the carbohydrates, in fact, are carbon with hydrate, that is water. And so if I take 5 formaldehydes, that's 5 times C1H2O5, I should be able to get 1 ribose. Ribose is the R in RNA. 2 glycolaldehydes and 1 formaldehyde, right, gives you 1 ribose. And by the way, it's been known for over 150 years that if you just stir some of these compounds like formaldehyde and glycolaldehyde in the presence of alkali, you actually make carbohydrates. This is a Russian chemist named Bularov who actually made sugars this way. Now, where do you get the alkali? Well, that's pretty easy. There is the so-called basalt. I brought a sample here. You look like an honest crowd, so we can pass it around. You're looking here at a basalt. Okay, so this is a basalt igneous rock. It's much like what you'll see in the highlands of Mars, but you'll notice the green rock that's inside the basalt, that's peridote, or olivine. It's also got a gemstone, which I have shown you here. When you put water and you erode that basalt, you generate alkali. And you generate alkali by this reaction called serpentinization. You make magnetite. hematite and magnesium hydroxide. And this alkali is what is known to get carbohydrates from formaldehyde. So you can say, hey, great. Butlerov in 1867 shown that working with that material, eroding it with a little bit of water, we ought to be able to get base and we can make ribose. But not so fast, and the reason for it's not so fast is because actually you make ribose, but then you destroy it as fast as you make it. And you have this horrible complexity. And I don't expect anybody to look at these structures. We've actually looked at all those chemical reactions and can tell you details about each one of them, but there are a lot of them. But for you, this is better known as the so-called tar paradox. Now the tar paradox is known with sugars in particular. Who's actually here grown crystals of rock candy? Raise your hand, yes. And then you take the rock candy as a nice substance, you then heat it for a while, and it starts to caramelize. It liquefies, and of course the more you caramelize it, in fact, if you continue to heat this, you don't get life emerging from this, right? What you do is get more and more tar, more and more asphalt, more and more stuff that's better appropriated for paving roads than starting life. So here you go. Unfortunately, RNA, ribose, is also capable of caramelizing. And now I don't assume that many of you are organic chemists in the room, but I just thought I would just give you a little chemistry. You got to look for this C double bond O carbonyl group, okay? The minute you have this C double bond O carbonyl group, you can have this reaction, which is called enolization. You can have this reaction, which is called aldol reactions, or an aldol addition, that regenerates a new molecule with a CO double bond O group, and then you can do this whole thing over again, all right? And so what happens, that's in fact where this complexity is coming from, is from that reaction. Those two reactions over and over again on molecules that have carbon atoms doubly bonded to oxygen atoms is what gives you these tars, and it gives you a real problem. And the reason why this is important for RNA is because ribose, there it is, has a C double bond O. And so ribose will be made in the Butlerov reaction, but it will be destroyed as fast as it's made. And so the amount that you actually get is not significantly large. And in fact, Stanley Miller, who's this great man who started modern prebiotic chemistry, in 1995 measured the rates of decomposition of ribose and other things, and wrote a conclusion, the implications for chemical evolution. And what he concluded was that stability considerations preclude the use of ribose and other sugars as prebiotic reagents. It follows that ribose and other sugars were not components of the first genetic material. And that's a real blow for the people who think that RNA was first, because if you don't have R, that is ribose, no R, no RNA. And so that's the problem in prebiotics. So here's what you've seen so far. Vitamins, the ribosome, all have RNA in them, and they all indicate that RNA is the first Darwinian biopolymer. In fact, we can make in the lab an RNA molecule that is Darwinian. We can get something that will replicate. Further, lightning, ultraviolet light in a primitive atmosphere gives you the precursors of RNA, like formaldehyde, as well as, by the way, of the RNA bases coming from hydrogen cyanide and cyanamide, which are other components of that mixture. So formaldehyde should give ribose, the last missing piece of RNA. However, ribose is destroyed. Okay, and that's your problem, by the alkali that's required to make it in the first place. So one of your solutions then comes from this guy. So I'll pass this around as well. This guy is a tourmaline. He's also formed in those basalts. It's also got a very nice gemstone. I have put a picture of it up there on the right. Peridot, this also has a lovely gem. These are great minerals for gems. And you got to keep in mind that minerals are there as well. You're not originating life in glassware, right? So sugars have these adjacent OH groups, more chemistry, sorry about that. Borate loves to bind to adjacent OH groups. There it is. Borate is found in that rock that is circulating around. And in fact, it's even better than that. When that rock erodes, it gives soluble borate salts, which then go into places like Death Valley and give you evaporite minerals like this one. This guy is culminite. It's a calcium borate. Very, very useful, because what ribose does is it binds to borate, and when ribose is bound to borate, it forms a cyclic structure which no longer has a C double bond O group, and it no longer decomposes in base. So borate is able to stabilize ribose and prevent its, you mind this, tarization. It prevents it from converting to asphalt. And just for the record, it constrains the reactivity to within this box of all of this. You actually make C5, 5-carbon sugars, a stable species if you got the green mineral in the basalt rock plus the white mineral that is eroding from the basalt to give you that, the black mineral is eroding to give you the white mineral in a dry valley. How are we doing? So there you are. Where, by the way, yes, can you find these minerals? Well, okay. In general, these borate minerals, other than tourmalines, are generally water soluble. So where you find them is in places like Death Valley, poorly named. But borate comes to the surface in these tourmalines. They're eroded with the peridot and basalt to give you these alkaline borates culminate, and those are the samples that are being circulated, and that gives you ribose. So what we've done, and I can, let me just see, is solve the very first problem that I've alluded to, and I'm listing these as paradoxes, right? The tar paradox, that is, you get tar out of sugars, well, not in the presence of borate. And it also turns out to solve the other paradoxes, that even if you can get ribose, it turns out that ribose in water falls apart. Water is corrosive. There's a phosphate paradox, which you heard about this morning. There may be apatite on Mars, that's this mineral, but it's not accessible. It's a precipitated solid mineral. And in fact, I've even brought you around a specimen of apatite so you can see how inaccessible that apatite phosphate is. If you try to get it out of there, you won't. If you eat it, you won't get phosphate. And by the way, the last problem or the last paradox is even if you make RNA, it's not stable enough to accumulate. So the desert actually, with borates, solves these other problems as well. So I mentioned how you can get formaldehyde into sugars if you have borate minerals with glycolaldehyde. I mentioned only in passing these other nitrogen-containing compounds. It turns out that hydrogen cyanide and cyanamide are also very useful, because they will react with formaldehyde to give this solvent, formamide, and this material, urea. Actually, you can make the RNA bases from these, but more important is that formamide, which has a boiling point of 220 degrees centigrade, is a liquid over its entire range, even at Martian temperatures. In fact, there can be liquid formamide on the surface of Mars at the time. But what's more important is that they're great solvents for making phosphorus, phosphate ester bonds in the backbone of RNA. So borate is a wonderful thing in the desert because it also prevents the phosphate from getting captured by calcium. So I mentioned calcium and phosphate goes to form apatite, which is this mineral now circulating, which is not giving you accessible phosphate. However, if you have borate present, what happens is that the borate with the magnesium captures the phosphate to give you this mineral called lunaburgite, and that leaves calcium and sulfate behind to form gypsum. So borate is rescuing the phosphate to keep it out of apatite, which is that mineral that's floating around you, and it actually makes the phosphate accessible. And we just can show you a couple of people. These are the two co-authors on the talk who actually do experiment. They take nucleosides, they take this lunaburgite, the magnesium borophosphate, and they show first that the borate is extracted by the ribose because there are two adjacent OH groups which coordinate to it. Then the phosphate from the lunaburgite goes, and it actually goes exactly one place, the one place that's free, the exact place that you want it to go to make RNA out of these materials. So you get two for the price of one. The borate is making the phosphate available in a desert until the nucleosides are present, and then puts phosphate on exactly the right place to make RNA. And so what you've seen so far is wonderful, right? You've got all these paradoxes, but borate now is preventing ribose from being destroyed under conditions where it's made. Borate is sequestering phosphate in the lunaburgite borophosphate to make it later available when ribonucleosides are available, and they form a phosphate exactly where they're supposed to as long as you have a desert environment with these solvents, formamide from hydrogen cyanide and urea from cyanamide. Now, this is kind of great because what we're doing here is By the way, there's also a way of making these bonds. I'm running out of time. But let me just point out that this is what we're building here is what we call the discontinuous model for the formation of RNA. There it is, replicating RNA, starting from carbon dioxide, water, dinitrogen, and a little methane, which goes by electrical discharge, formaldehyde, hydrogen cyanide. It makes the ribose, which is stabilized by borate minerals. You can then put the phosphate on in a desert environment, and you can make a oligomeric RNA. And in fact, this entire piece a spectrum has, this entire scheme has experimental data for each steps. Now, by the way, the last question is, of course, even if you make RNA, it's not stable, how do you keep it? Well, it turns out that minerals help this also. This is work that Lisa Biondi has done, just showing how minerals, especially high atomic weight barium carbonate minerals, will bind RNA and stabilize it and form a stable product. So this means that the four paradoxes I mentioned, the tar paradox is solved by borate minerals in a desert environment, which prevent tar formation. The water paradox, which prevents us in water from forming phosphate esters, is solved by borate minerals in a desert environment that allow unstable phosphate bonds to form. The phosphate paradox, meaning it's hard to get phosphate, borate minerals in a desert environment deliver phosphate, and once you've made the RNA, you can capture that on a desert environment. So that's what you've seen so far. But now what about Mars? So the question now is, were these minerals and deserts present on early Earth? Well, good question. Bob Hazen over here at Carnegie and Ed Grew up in Maine say no. Joe Verschink says we don't even have deserts. We have this back and forth between the chemist and the geologist. And so our work has caused geologists to look again, meaning that they can actually find sediments that contain borate minerals metamorphosed out of 3.8 billion year old sediments from, for example, Greenland, also in Australia. So we can find borates at 3.8 billion, but Joe Kirschvink has now said, no, we may have the borate minerals, but we don't have the deserts. And the reason for that is because the models for planetary formation say that the inventory of water on Earth is so high, and plate tectonics has not yet started, so the elevation differential is so small that all of Earth was a Kevin Costner water world. And you actually heard some of that in some of the talks this morning as well. So Joe's solution, our solution, is to take all of that chemistry to Mars. Why? Because Mars has never had the level of water that Earth has had in its inventory. And therefore, you've always had dry land on Mars. You've always had an evaporation cycle that is required to get these evaporate minerals, like that white mineral that's now circulating among you. And so the logic tree becomes as follows. First, you have to decide whether RNA was the first Darwinian molecule. Now, vitamins, the ribosome all say yes. If you say no, you have a different set of problems that we don't talk about today. But if you say yes, then you need to be able to make RNA prebiotically. So then you have to say, well, you're going to use Benner's stupid chemistry. And if you say no, my response is, get your own goddamn chemistry. But if the answer is yes, you need a desert. You need oxidized minerals, as I've shown you, in what's circulating. So now you have to ask the question if you can say, well, can we have those on early Earth? Well, if the answer is yes, then life originated on Earth. But if the answer is no, then we are all Martians. That is, when we go to Mars, we will be going home. So that's all I have to say. One thing I think I should make emphasis is most people I talk to don't understand how far models have advanced for our understanding how life originated. Right? Many people think this is one of these great unsolvable problems, unapproachable by scientists. That's really not the case. I mean, we have a relatively coherent model for a dry valley. The only real question you have to ask is that a desert on Earth or is it a valley on Mars? I must say that, as I've mentioned, we do sell origin of life jewelry because this is a very attractive set of minerals to be working with. That's www.fame.org. You can go back and look at. But with that, let me stop. Thank you for your attention. I think I might have a few minutes for questions.
Do you have time for a few questions?
Yes, sir.
Is it possible to be statistically coherent about saying something like penguins didn't spontaneously evolve, like just kind of appear in Antarctica, they probably came from somewhere else. The last universal ancestor is quite a high number of different biochemical processes going on. arguably, since none of its cousins appear or something, kind of going back to these antecedents you talk about, they might be somewhere else? Or is it possible to prove they haven't been destroyed?
You've lost me on the penguins part. So first, yes, penguins went to, 30 million years ago, 35, 40 million years ago, in the Eocene, the Earth was about 17 degrees warmer than it is today. There was no sea level ice anywhere on Earth, including Antarctica, which had a deciduous forest. The Earth has had a planet. I mean, for those of you who are worried about global cooling, global warming, the historical pattern on Earth for the last 35, 40 million years has been global cooling. Antarctica frost over. So that's how the penguins got there. Now, what was the other question?
So we've got this last universal ancestor.
Yes.
You can call up Roche tomorrow and they'll send you a big poster full of all the
Yes, they will.
Yeah. Everything on Earth shares. And
yes.
It looks much more like something that came here from somewhere else.
Yeah.
It's kind of worked its way up.
Exactly. So the point is that the last universal common ancestor is an organism that looks very much like us metabolically, as long as you're willing to make me single cell. So it looks very much like a modern bacterium. And so the problem now is dating that. Well, the answer is that's going to be dated roughly 3.5 billion years. There's a large error bar associated with that date. Now the problem is that's a billion years this side of the origin of the planet, right? So you have more, and there's more time between the origin of Earth and the dating of that Luca than there is between the emergence, separation of you and corn and today. So a lot has gone on lately, and we have this big billion years where we don't know whether that's a lot of time to get stuff that's complex like Luca or too little. Now the usual argument for panspermia, right, is that it's too little time to get Luca, and therefore we had to have more time on another star. And all I can tell you is that, you know, your star is going to become a red giant and make this place uninhabitable in a billion years. You have waited 80% or 85% of the, maybe 90% of the useful life of this star to get to the marvelous state of biology that you represent. You will either have to go back to Mars to escape the sun as it expands or not. But that indicates that it's unlikely for you to get to your state, but the argument here is it's very likely to get to a microbial state. I haven't answered your question, but there's a lot of time up front to get complexity at the metabolic level. What seems to be the case is it's hard to get multicellularity compared to getting metabolic complexity.
That was a really thorough answer, so we're going to go ahead and move on from this speaker right here, but thank you very much.